We got just a little bit more to talk about the male side, and I put in a few slides from my own stuff that are just really illustrations more than anything else. But we were talking about spermatogenesis, and I had drawn my lovely picture of a seminiferous tubule, but I've got some images to show you of that. But this is all under the, in terms of endocrine regulation of spermatogenesis. It all starts up in the brain for both sexes with the same hormones. Gonadotropin-releasing hormone from the hypothalamus, which stimulates the anterior pituitary cells to secrete FSH and LH. And in the male, the FSH stimulates the process of spermatogenesis, and the LH stimulates Leydig cells, which are in between the seminiferous tubules to produce testosterone, which is critical for the process of spermatogenesis. This is an image that I put in over the weekend of the testicle, showing a cross-section of it. And what you're seeing here, and I wanted to point out, is the extensive seminiferous tubules that are all throughout both testicles. And if you took the seminiferous tubules and stretched them end to end, they'd be about the length of 3 football fields. So it's quite extensive in terms of surface area. And the process of spermatogenesis is occurring at every section along that 3 football field length. So it's occurring extensively once the male hits puberty, obviously. But that is why there are the ejaculate is so highly concentrated in sperm cells, because they are coming at all lengths of the seminiferous tubules. And then when the sperm get to the actual tubule, they detach and are washed down the tubule and end up right here in the epididymis, which is just a continuation of the tubular structure, and it's on the posterior aspect of both testicles, and that's where the sperm are stored until ejaculation occurs. You can see right here that all the seminiferous tubules are all merging into this structure called the ret testis, and from the ret testis they enter into the epididymis. And at ejaculation, we're going to find out that they are propelled up into the abdomen through the vas deferens, or ductus deferens. This is another cross-section looking from the top down, and you can see how the seminiferous tubules are compartmentalized into lobules by tissue called septa. And you can see there's on average of 4 to 5 lobules per testis. That's not a fixed number. And then this is just a microscopic actual micrograph of the testicular tissue. Make that go away. And what you're seeing here are the individual lobules, and they drain into the ret testis up here. And then this is an image of what I was drawing over there. Again, if you look at testicular tissue under the microscope, it's wall-to-wall, side-to-side, top-to-bottom seminiferous tubules. And what you have on the periphery of each seminiferous tubule are the germ cells, which give rise to the sperm eventually. These are the diploid spermatogonium, and they initiate the process of meiosis through initially undergoing mitosis. And one spermatogonium will divide mitotically into 2 identical cells, and one of those spermatogonium remains in the periphery of the seminiferous tubules and maintains what's called the spermatogonial pool. So in the male, we never run out of the germ cells that give rise to these cells. And the other spermatogonium then starts the process of meiotic cell division, reduction division, to eventually become that haploid sperm cell, okay? And that process of meiosis occurs through the tissues of the seminiferous tubule until it gets right to the edge of the lumen of the canal, if you will. And again, at this point, once meiosis is complete, they're called spermatids, but they look like cells. They look like a round cell. And it's not until meiosis is complete, there's a process called spermiogenesis where they morphologically change into that round spermatid into the mature sperm cell. And if you look at this on the computer screen very closely, this is probably a 100x magnification. You can just see the heads of the sperm and actually look at some of the tails. They back up into the lumen, so they're like going in reverse. But wherever you see tubules that are kind of darkened a little bit, like this one, it's full of sperm cells, whereas the one over here is not devoid of sperm cells, but very few. And so this tissue is reproductively active. And actually down on this one, you can really see the sperm heads and the tails. But nonetheless, that process occurs from puberty until the life of the individual is over. It continually is processed because FSH is continually processed and LH is continually processed, so testosterone is continually processed. So it's spermatogenesis itself is a continual process. And this is just an enlargement of that micrograph showing just one individual seminiferous tubule. And you can actually see some of the cells up here mitotically and meiotically dividing because of the condensation of the chromosomes. And you can also see down here, these are the actual heads of sperm cells. And if you look really closely, you can see their tails. And then they're going to wash down through the lumen, eventually going to end up in the epididymis, that structure behind the sperm cells. And this is just an illustration of that process, looking at one section of the testicular tissue, starting with the spermatogonium, and how they undergo the process of meiotic cell division and spermiogenesis. And it's also important to understand, again, I highlighted this last week, I believe, is there's a very important structure that's formed in part by these nurse cells. They're formerly known as Sertoli cells, and they form what's called the blood-testis barrier. Because you can see this is vascular tissue. Here's a capillary right here, very close to the developing sperm cells. And through the process of meiosis, regardless of if it's sperm or egg, genetic recombination, genetic variation is occurring. So these cells are genetically different from the host cell. And if it weren't for this blood-testis barrier, our immune cells would identify the sperm as foreign cells and mount an immune response. And if that blood-testis barrier is ever compromised through testicular injury, that does happen. And B cells will produce what are called anti-sperm antibodies. And it's a form of subfertility or infertility in the male. This is another image I put in showing you the anatomical structure of the sperm. So after spermiogenesis, this is what they look like. They look like they know what they're doing. They got all the pieces and parts, the head, the acrosomal membrane, the midpiece, which is full of mitochondria, because this is the gas tank, if you will, and then, of course, the tail. But at this point, even at the initial process of ejaculation, they don't have fertilizing capacity. The image on the left is a transmission electron micrograph showing you the ultrastructure of the sperm cells. You can actually see the mitochondria. The head of the sperm is always a very dense structure, just simply because it is filled with the DNA. And then on the top of the sperm cell is a very important acrosomal membrane, which we'll get into in just a minute. This is also a transmission electron micrograph, and this is the tail of the sperm, showing that the tail is composed of microtubules, the same microtubules you learn about in the cytoskeleton of cells, the large cytoskeletal fiber. And in the tail of the sperm, they're arranged, coupled, two microtubules together, and they form what are called doublets. And they are arranged such that there are 9 doublets wrapped around a centralized pair. And it's through the interaction of these doublets that the sperm swim. If you look in between the doublets very closely, you can see these shadows. And these shadows are called motor proteins. Specifically in the sperm tail, they're dynein molecules. And what happens is, under the influence of ATP, those dynein molecules will bind to the adjacent doublet pair. That's redundant. The adjacent doublet, and pull on it, just like this. And at that microstructural region, that's all that's going on. If you imagine my hand is one of the dynein molecules and this is a microtubule, it'll bind and pull. And it's very much similar to how actin and myosin bind to each other and interact when skeletal muscles contract. Now, the sperm cell's not contracting, but when you have this interaction repetitively down the length of the sperm tail, with all of 9 doublets and the central doublet interacting together, it causes the tail to whip back and forth, and the sperm can swim. And interestingly enough, from an evolutionary standpoint, when you take a cross-section of any flagella on any organism, an amoeba or cilia that don't have a function in motility per se, they're all arranged like this. That 9 doublets wrapped around a centralized doublet pair. Makes you think a little bit about evolution, but this is not an evolution class, so we're not going to talk about that. This is this is from your book, a depiction of the hypothalamo-pituitary gonadal axis. And when this hypothalamo-pituitary gonadal axis is established, that defines puberty. Okay, that defines reproductive capability of the individual. And I know we talked about this last time because we were talking about this kisspeptin, but the activation of GnRH on the anterior pituitary and its effect on FSH and LH, the testosterone source, and the hormone required for spermatogenesis, that process continues or is initiated. We also talked about the negative feedback effect of testosterone and inhibin on the hypothalamus and anterior pituitary, but like we said, it's a very weak negative feedback mechanism, okay? Meaning that testosterone is produced all the time. The amount of testosterone production is fluctuated based on this negative feedback mechanism, but it's never completely inhibited like we'll see in the female. Okay. I think we talked about all this just for reference sake, and I think we talked about this last time, but a vasectomy is an actual ligation of the vas deferens. So testosterone still occurs. We have the reproductive functionality of the male. It's just the sperm can't get to where they need to go because there has been a excision of a portion of the epididymis or the vas deferens. Okay. We also talked about the importance of the accessory sex glands, the 2 seminal vesicles, the prostate and the bulbourethral gland. Remember, the seminal vesicle produces the majority of the fluid of the ejaculate, about 60% of it. It's got some very important components in it, like the fructose, the carbohydrate required for cell respiration in the sperm. And so this is akin to the gas, filling that gas tank, filling that mid-piece with all the mitochondria in there. It's also got a lot of other stuff. These are just some of the important ones. The fibrinogen, which causes the agglutination of sperm initially. Seminal plasmin, which is a naturally occurring antibiotic. Then the prostate, where the sperm are actually combined from the two testicles, right where the two vas deferens meet at the what's called the ejaculatory duct, right at the anterior, the posterior side of the prostate. And you get the contribution of prostatic fluid. Some important components of that are an enzyme that's going to eventually break down that fibrinogen agglutination reaction. It's also got alkaline buffers to buffer the prostatic urethra and the penile urethra to an extent. And then lastly, the bulbourethral glands, which are small glands and the last of the accessory sex glands, and this also secretes an alkaline fluid to further buffer the penile urethra and potentially the vagina as well. And seems like there was something else I wanted to say, but can't remember. So this table gives you the all those components. Particularly, it's important. There's more of a description of the accessory sex gland fluids. That is very informative. And lastly, in terms of the male sexual act, you know, what's going on in the process of the sex act in terms of anatomy and physiology in the male. It requires, obviously, an erection, and the penis itself is composed of 3 different layers of what's called spongy tissue, and really, microscopically, it looks like a sponge does. And what happens is when erection occurs, well, in a non-erect penis is mediated, or the sympathetic nervous system predominates. And what the sympathetic nervous system is doing is causing vasoconstriction of the arterioles that supply this spongy tissue with blood. And so it's limiting blood flow. And again, that's the sympathetic nervous system. Remember, we talked about how the sympathetic nervous system, fight or flight, it's revving up those components that we need for the fight or flight response. We're not necessarily worried about reproduction at this point, so it is maintaining in this case, a flaccid penis through the vasoconstriction of these arterioles. But then when the stimulus occurs, through it says stimulation of mechanoreceptors, not necessarily. There can be thoughts of sexual acts and arousal from that standpoint. So it's not specifically through mechanostimulation, but what happens is now the parasympathetic nervous system predominates and causes the vasodilation of those arterioles supplying blood to the penis. So the penis gets engorged with blood, and that's what an erection is. So it's all through the autonomic nervous system, both sympathetic and parasympathetic. And now every time you turn on the TV, there's a new commercial about erectile dysfunction and treatment of that. That has been a financial boom for the pharmacological industry because these medications are very effective. And what they do is specifically mediate or inhibit the nitric oxide pathway. Specifically, what most of these medications are a cyclic GMP specific phosphodiesterase type 5 inhibitor. And what phosphodiesterase is an enzyme that breaks down cyclic GMP, and cyclic GMP is another type of second messenger, very similar to cyclic AMP, just a different nucleotide. Okay, and through the natural process, through the parasympathetic innervation, we have the activation of second messenger systems that ultimately cause the vasodilation of these arterioles. And cyclic GMP is the second messenger. When it becomes phosphorylated, it becomes active, okay? And you get the vasodilation and you get the erection. And this medication is an inhibitor of that enzyme. You inhibit that enzyme and you take out that effect, and you have vasodilation for as long as the medication is biologically active. And interestingly enough, y'all might even know this, but Viagra was originally marketed as a treatment for heart disease because of its vasodilatory mechanism. But they found that it had another side effect, a positive side effect, depending on who you are. And so it was marketed then for that, and has been just a boom in the pharmacological industry because it's specific for this phosphodiesterase type 5, but that's not to say it doesn't stimulate other phosphodiesterases, because what Viagra is a high concentration of this inhibitor. And if you hit anything with a high enough concentration and it's a similar biochemical interaction, it's going to affect all kinds of other systems. So it's effectively a phosphodiesterase inhibitor. It's just specific for type 5, but if you hit these other phosphodiesterases with a high enough concentration, it's going to affect those too. So what Viagra does is cause systemic vasodilation. And that's why they tell you, you know, when you see these drug commercials, at the end of it, they'll go through all the secondary negative side effects, from subtleties to death, you know. And one of the contraindications, if you're on blood pressure medication, you shouldn't take this medication because it can further enhance that decrease in blood pressure through vasodilation. Nonetheless, so this is the neural control of excitement and erection. Let me move this out of the way. Okay. So again, it starts up in the brain because we're talking about the autonomic nervous system. The sympathetic nervous system is predominating when during a non-stimulatory period, if you will, and specifically it's causing vasoconstriction of these vessels, so limiting blood supply into that spongy tissue of the penis. But with stimulation, whether it's mechanical stimulation or otherwise, the parasympathetic nervous system is going to take over, causing vasodilation of the arterioles and erection, okay, and also causing, to a degree, secretion of the some of the secondary accessory sex glands, particularly the bulbourethral gland, and this is prior to ejaculation. And I think I mentioned last time that you've heard of the term pre-ejaculate. That's coming from the bulbourethral gland, initially through stimulation from the parasympathetic nervous system and the preparation for the process of ejaculation. Ejaculation and emission are two synonymous terms, but it can be further broken down into emission and expulsion. The process of emission is the two segments, if you will, emission and expulsion, collectively are ejaculation. Emission is the early sequence of events that is ejaculation, where the sperm leave the epididymis up through the vas deferens and get to the ejaculatory duct within the prostate, okay, the prostatic urethra. And then expulsion is just that, the elimination of that ejaculation from the body. This is all part of the orgasmic response, and it tells us here that orgasm gives us a feeling of release and feeling of deep relaxation. This is all, at this point, these emotions or feelings or whatever you want to call them, this is all due to oxytocin. Oxytocin levels are dramatically increased in both sexes at orgasm. Okay, and there's a reason for that, because what oxytocin does, we learn about oxytocin a lot when we talk about parturition, the process of birth in the female, and how it causes those very strong uterine contractions. What oxytocin does is cause smooth muscle contraction. And its benefit in the male, in terms of a physiological standpoint, is to cause smooth muscle contraction of the urethra. That's what the urethra is, it's smooth muscle. And it causes muscular contraction and a wave-like, peristaltic-like contractile response in the penile urethra to further help propel the sperm out. Because even though ejaculation has occurred or is occurring, and the sperm at this point have already been introduced to the fluids from the, for sure, the seminal glands, and again, the seminal glands contain the fructose, so they can swim at this point, but they really don't, because they're in that agglutinated clump. And so the peristaltic action of the urethra, and it also continues on in the vagina as well. And there's prostaglandins that come from the uterus and vagina that further help that smooth muscle contraction to further assist that journey of the sperm cells. Okay, volume and sperm concentration vary through multiple ejaculations in a short period of time. The sperm concentration is going to decrease. So if pregnancy is something the individuals are striving for, they want to space out copulations, and actually to enhance the process of fertilization, there should be copulation about 1 day before ovulation occurs, because you want the sperm, and we're going to see when we get into the female tract, we're going to see where fertilization occurs, but it occurs very high up in the oviduct. So you need the sperm already there waiting on the process of ovulation to occur. But volume and sperm concentration is not really something we need to worry about. Unless you're working on a breeding farm, but we're not going to be doing that, are we? Horse breeding farm, that is. Okay, so that's the male. Any questions about us fellows? Nope. Female. Female reproduction is, again, in my opinion, much more interesting, because it's a very dynamic process and all kinds of different things going on. These are two images from my lectures, and I just thought they were really cool. The one on the left is a MRI of obviously a pregnant female close to term. You can see the head of the fetus is right where it's supposed to be, right on top of the cervix, pushing down. And this is what initiates the process of parturition. And the image on the left are on the right are two blastocysts. And we'll talk about blastocysts in a minute, because this is the stage at which this fertilization has occurred. These cells are about 5 days old, and this is the stage at which they implant in the uterus. So between the time of actual fertilization and implantation of an early embryo is about 5 days. Okay. And these two blastocysts, you might have already seen this if you looked at the slides earlier, but one of these is my son Christopher, because he he was produced through in vitro fertilization. And I always joke about how when I first saw this picture, I wanted to know if there was any family resemblance, you know. And so I looked really close and I saw this. I don't know which one is Christopher. Obviously, one didn't take, but that's a blastocyst, and this is Christopher's first baby picture. Just not sure which one. The steps in oogenesis is similar to the male in that it is meiosis, but that's where it stops, because there's some inherent differences between oogenesis and spermatogenesis. It starts with the germ cell, an oogonium, as opposed to the spermatogonium, obviously diploid cell. First division is mitotic division, but then that's where it ends in terms of a comparison between the two, because oogenesis begins prior to birth. The first mitotic division and the initiation of the actual meiotic division begins at up to about 5 months of gestation. So a 5-month-old fetus. They have what are called in the ovary primordial follicles that have been arrested in development at about, the initiation of meiosis occurs about the 7th month. But then what she has is what are called primordial follicles occupying the cortex of both ovaries. Okay, so the female, the human female, well, mammalian female, I should say, at birth has all the follicles, which is to say all the eggs she's ever going to have. By the time birth occurs, she has on the order of 1 to 2 million primordial follicles, so 1 to 2 million potential eggs, but that number is drastically reduced by the time of puberty to about 500,000. And from that cohort, actually about 400 to 500 will actually be ovulated. But even though she has a finite number, she has all the follicles she's ever going to need. Okay. But again, those primordial follicles are arrested in development prior to birth and aren't initiated again until puberty occurs, until that hypothalamo-pituitary gonadal axis is established. Okay. And in the primordial follicles consists of the actual egg, and it's referred to as a primary oocyte in the primordial follicle. But it's the egg itself and a single layer of a cell type called granulosa cells. And the granulosa cells, as she becomes reproductively active, are going to start producing an estrogen. And they are producing estrogen at this point, but trace amounts. Okay, and then as the follicle furthers development after puberty has occurred, and we'll see some images of these structures, it transitions into a secondary oocyte that's in a tertiary follicle. It gets kind of complicated. When I refer to the egg, I just refer to the entire follicle, and the follicle is the egg itself and all the associated cells and structures. Okay. But then that secondary oocyte in the mature tertiary follicle is the stage at which ovulation occurs, and it's released into the oviduct. Pubertal changes in the female, similar to males. It happens a little earlier, age-wise, in the human. But again, what defines puberty is when the hypothalamus starts talking to the anterior pituitary, it starts talking to the gonads. Once you have that communication link, puberty has occurred, and that individual is reproductively active, reproductively mature. Okay. This is what it looks like in the female. Again, it all occurs, or begins, I should say, prior to birth with mitotic cell divisions, where they're arrested in development, until puberty occurs. You can see they're still diploid, but they are, these images are just the egg itself. There's also a layer of granulosa cells around each. But they are arrested in development in the ovary, specifically in the cortex of the ovary. And when puberty occurs and we start to get the production of FSH, follicle stimulating hormone, this is where that hormone gets its name because it stimulates the development of the ovarian follicle. Okay? And then that follicle is going to undergo changes, and again, the picture here is just showing you the egg itself, so it looks the same all down the way because the egg doesn't change. Okay? The structures around the egg, the cells and structures around the egg change. And also a big difference in oogenesis as compared to spermatogenesis is that oogenesis is never complete unless fertilization occurs, because the interaction with the sperm cell is what initiates the last meiotic cell division. So unless fertilization occurs, meiosis is never complete. Okay? And once we have fertilization, then we're going to start the process of mitotic cell division. And you can also see here, once we start getting the meiotic cell division, we have the emergence of these things called polar bodies. Remember what's happening in meiosis, reduction division. We're reducing the chromosome number, right? And so in the first meiotic cell division, we are separating the sister chromatids, right, after DNA synthesis has occurred. And then in the last meiotic cell division, we're actually separating those sister chromatids. Y'all with me on that? Yes? No? Yes? And remember in the male, in spermatogenesis, we go from one spermatogonium into 4 sperm cells. Okay? That doesn't occur here because when the meiotic cell divisions begin, we're going to end up with one cell that's going to continue through oogenesis, and the other one is referred to as a polar body. And that's that second set of chromosomes, and that's absorbed by the body. And so you see there's 2 emergences, is that a word? Of polar bodies. Through the first meiotic cell division, we get the cell and a polar body. And then in the second meiotic division, if fertilization has occurred, we get the emergence of the second polar body. And these aren't anything that's detected, except that the emergence of that second polar body is something they look for in vitro clinics. Because when you put sperm together with an egg and you're hoping for fertilization, if you see the emergence of a second polar body, and you can see it microscopically, if you see the emergence of that second polar body before any mitosis occurs, you know that fertilization has occurred. If you sat there and looked at it long enough and fertilization occurred, you're going to see the cells start dividing. And one cell becomes 2, becomes 4, becomes 8 within that, what's called a zygote at this point. But if you're looking for it and you see the emergence of a polar body, you know fertilization has occurred. Okay? So at the end of oogenesis, we're only going to end up with one oocyte. Okay? And this is just side-by-side spermatogenesis, where we end up with 4 sperm cells, but oogenesis, we are going to only end up with 1 and 2 polar bodies. That's supposed to be an arrow. Okay? Now, this is all under the control of hormones. Okay? Completely different from the male. It starts out similar with the GnRH from the hypothalamus, FSH and LH from the anterior pituitary, but then it gets completely different because the female is programmed for 2 separate events through the reproductive cycle, and that's follicular development on the front side, ovulation right in the middle, and then preparation of the female for pregnancy and implantation of a potential early embryo on the second half of the reproductive cycle under hormonal control and completely different things going on. Okay. The human female also undergoes menopause, and that's only specific for the human, believe it or not. You would think that at the very least, you know, lower primates, female primates, lower primates, the monkeys and the apes, would undergo something similar, because genetically we are very similar to those animals. There's only about a 5% genetic difference between us and them, but they don't undergo menopause. That's only for the human, and obviously that's when reproductive action ceases. So the ovarian cycle is subdivided into what's called the follicular phase and the luteal phase, with ovulation right in between the two. The follicular phase is called the follicular phase because it's the phase where we're developing that ovarian follicle, taking that primordial follicle and stimulating it to undergo development. Actually, there's a handful of primordial follicles that initiate development. But then we have ovulation of, usually in the human, one follicle. Sometimes there's two. Sometimes there's more than two. But then after ovulation, the luteal phase. And it's called the luteal phase because a structure called the corpus luteum predominates, and that is the initial source of progesterone that is going to further aid in the development of the female reproductive tract for pregnancy. Okay, in the human female, this is about 28 days, give or take. Ironically, it's the same amount of time in the horse. The mare has a 28-day cycle. But again, the follicular phase is development of that follicle. Again, the egg itself stays the same. It really doesn't change, only if fertilization occurs. Now, it does change in terms of meiotic cell divisions, that kind of thing, but it doesn't change in size or shape or anything like that. And it goes through stages of development. And are you raising your hand or stretching? Okay, that's what I thought. The initial stage of development is called a preantral follicle. Preantral, before the antrum, and the antrum is a fluid-filled cavity that we'll see in just a minute. And the antrum gets bigger and bigger and bigger and bigger as the follicle develops. And the follicle is developing much like a water balloon, getting bigger and bigger and bigger before it explodes. Okay. Through this process, this initial follicular development, we're going to get an increase in the amount of estrogen produced. So if you looked at the estrogen profile, and we are going to see an endocrine profile towards the end of this, but if you looked at the first 14 days of the female in terms of endocrine hormones, estrogen is continually increasing up until the time of ovulation. That's because that estrogen, if you remember, is coming from those granulosa cells. In that primordial follicle, we had just the egg and a single layer of granulosa cells. And as that follicle is developing under the influence of FSH, those granulosa cells are mitotically active. So we're increasing the number of granulosa cells around the oocyte. And if they're producing estrogen, you can imagine the estrogen profile or the estrogen concentrations are continually increasing. At this point, there is no progesterone because the progesterone comes from that corpus luteum that we don't see until after ovulation. So estrogen levels are high, okay? And the FSH levels are increasing, not to a degree that you see the estrogen or the LH prior to ovulation, but nonetheless, estrogen is increasing. And estrogen and progesterone work hand in hand in preparing the female for pregnancy. One of its biggest effects are, through both of them, their action is on the not the myometrium, but the endometrium of the uterus, and it's getting thicker and thicker and thicker and more vascularized. making a conducive place for implantation of the early embryo. Okay, but another important effect of estrogen is that what's going to cause ovulation is luteinizing hormone from the anterior pituitary. And what we see from an endocrine standpoint is right before ovulation, right before mid-cycle, so about day 13, you see this very prominent, in the human, it's different in other animals, but a very prominent what's called LH spike or an LH surge. And that LH causes rupture of the ovarian follicle. Another effect of estrogen, while it's increasing prior to the LH surge, is it stimulates the follicle to upregulate LH receptors. So that's going to be sensitive to LH when it occurs. And we talked a bit about hormonal interaction. If you remember, one of the interactions was permissive effects, where one hormone is necessary for the action of another. And this is an example, because estrogen has to be there prior to LH. Otherwise, the LH is not going to be effective, because there won't be any LH receptors. Okay. And we also talked about synergistic interactions of hormones, where two hormones, they can do the same thing, but together there's a more profound effect. And the effect of estrogen and progesterone, they have synergistic effects on the endometrium of the uterus. Okay, so this is from your book, and this is the development of the egg. Again, the wording is so small you can't see it. Why does this do this? It stops writing after a while. Ugh. I didn't mean to do that. Yeah. Okay. Starting up here with the primordial follicles, these are all present at birth and up until the time of puberty. And they are composed of the egg, central portion, and the single layer of granulosa cells. And then at puberty, when the hypothalamo-pituitary gonadal axis is hooked up, then we have the development of this follicle, and you can see the egg is still the same size, but now we're getting more granulosa cells, and also another cell type called theca cells. And then as the follicle transitions into what's called the secondary follicle is characterized, again, by the egg itself, more and more granulosa cells, and the emergence of this follicular antrum. This little yellow portion and all the little spaces here are the emergence of that follicular antrum, and what they're going to do is eventually fuse into a great big one. And I put this in this morning. This is a scanning electron micrograph of a secondary follicle. And you can see the multiple layers of granulosa cells and the emergence of that follicular antrum, which you can see is just an open space. It's not open, it's filled with fluid, okay, and the egg. Okay. And then as that follicle matures, you can see the follicular antrum gets bigger and bigger and bigger until at this stage it's called a tertiary follicle. It's also called a Graafian follicle. I'm sure that word's in here somewhere, but I can't read that small. And it's at this stage that the follicle will rupture once LH is released. And LH binding to its receptor and causing a literal breakdown in the ovarian and the follicular wall, coupled with the increase in size of the follicular antrum, because that continues to increase the whole time, and it's like a water balloon. And that effect of LH on the outside of the follicle, coupled with the increase in hydrostatic pressure from the follicular antrum, the follicle bursts. And what you see if you looked at an ovary of a female that's reproductively active, you'll see all these little bumps, if you will, all along the follicular or the ovarian wall, and those are ovarian follicles bulging out from the ovary, and it causes just that little touch of LH coupled with the increase in pressure in the antrum, and it bursts and releases the egg into the oviduct. Okay, and then what happens is we remain behind in the ovary. The remnants of that ovarian follicle form what's called the corpus luteum. And what we have is we still have a layer of granulosa cells, so we're still having estrogen production. But this corpus luteum, and corpus luteum is Latin for yellow body, that produces progesterone. Okay, E2 and P4 are my abbreviations for estrogen, not mine, but the abbreviations for estrogen and progesterone. And that progesterone continues. That corpus luteum has a finite lifespan of about 5 to 7 days, so the source of progesterone is there for about 5 to 7 days. If pregnancy occurs, a process called luteal rescue happens, where the corpus luteum is salvaged, so the source of progesterone continues for a longer period of time until the tissues of some of the tissues that form the amniotic sac take up progesterone production through the remaining time of gestation. But if pregnancy doesn't occur, the corpus luteum degenerates into a structure called the corpus albicans, and the corpus albicans doesn't produce anything. And so the source of progesterone is lost, the granulosa cells eventually degenerate, and so the source of estrogen is lost. There's also another hormone that comes from the corpus luteum, that's inhibin. We saw the same hormone in the male. And so once the source of progesterone and estrogen are lost, the corpus luteum degenerate in the corpus albicans, the granulosa cells degenerate, those two hormones drop off, and that's the biochemical cue for the female to start the cycle over. And in the human, the first part of the cycle, the first 3 to 5 days, is menses, where she will shed the endometrial lining that's been developing under the influence of progesterone and estrogen. And then the process starts anew with FSH and eventually LH. Okay. I put this in, I think yesterday, and this just shows you those developmental stages in a active ovary. Again, all along the cortex of the female ovary, again, prior to birth, is filled with primordial follicles. Follicles that have been arrested in development in early meiosis, and with the advent of puberty and the establishment of that hypothalamo-pituitary gonadal axis, under the influence of FSH, a cohort or just a certain number of those follicles will begin to develop. And what stimulates a certain number of follicles to develop while follicles right next door to them don't develop is a big mystery. But nonetheless, a group of follicles begin the developmental process, going from primordial follicle to a primary follicle, and from a primary follicle to secondary. And sometimes under the microscope, it's a little bit difficult to differentiate a primary from a secondary. Tertiary follicles are always easy to identify because they got these great big follicular or fluid-filled follicular antrums, multiple layers of granulosa cells, and they're literally bulging out on the ovarian wall. Sometimes it's a little hard to differentiate primary from secondary, but once you see the emergence of these fluid-filled spaces, that's a secondary follicle. Okay, you can hear like somebody refer to a early secondary or late secondary or late primary. It doesn't matter. Secondary follicle is secondary when you see the emergence of those little fluid-filled spaces. Okay, and then under the influence of LH, we have rupture of the ovarian follicle. The egg itself goes off into the oviduct, which is this reddish tissue around it. Okay, that goes off into the first section, which is called the infundibulum, And then the remnants of that ovarian follicle differentiate into the structure called the corpus luteum. And the corpus luteum produces progesterone. So we don't see any appreciable progesterone until the post-ovulatory period. And if pregnancy doesn't occur, that corpus luteum degenerates in the corpus albicans. Again, you can see it's just that white tissue. It doesn't produce anything. Progesterone source is lost. The granulosa cells degenerates. The estrogen is lost. And that's a biochemical signal to start the cycle over again. These are some images I put in again this morning. This is a good one showing a ovarian section that is rich in primordial follicles all around the cortex, and we know this individual is reproductively active, or post-pubertal, I should say, because we're looking at the emergence of follicular development. Okay, primary follicles, the egg itself, and associated granulosa cells. This is a table from your book describing the differences in the follicular stages, both anatomically and from an endocrine standpoint. But the importance is to understand the follicular development is occurring all under the influence of FSH. The process of ovulation is all under the influence of LH, coupled with increase in pressure from the follicular antrum. Okay, and then after ovulation we have the luteal phase, and again it's called the luteal phase because the corpus luteum predominates, and this is the structure that produces progesterone. And even though, and we'll see in a minute when we look at the endocrine profile, we have the increase in estrogen through the first half of the cycle, and then ovulation occurs, and the estrogen levels do drop significantly from their peak concentration prior to ovulation, but we still have granulosa cells that are associated with the corpus luteum, so we still have estrogen concentrations. They're not as high as progesterone, but those two hormones stay high for the majority of that luteal phase. But again, it's referred to as the luteal phase because that corpus luteum predominates, and the hormones estrogen and progesterone, and now inhibin, predominate. Okay. This ovarian cycle is tightly regulated. If you remember in spermatogenesis, we talked about the negative feedback pathway and the inhibition of LH and FSH and GnRH from testosterone and from inhibin. And we said it was a weak negative feedback pathway. It's a very pronounced negative feedback pathway in the female. Again, because the progesterone and estrogen and inhibin collectively significantly inhibit the production of LH and FSH, because in the latter half of the cycle she wants to concentrate all her resources on preparing for pregnancy. She doesn't want to waste any time and energy, any expense in follicular development, so those three hormones significantly inhibit FSH and LH and GnRH so that they are baseline through that luteal phase. So we can concentrate all on preparation for fertilization and implantation of an early embryo. Okay. And so through the whole 28-day cycle in the human and the horse, except for menses. Menses, the menstrual period, menstrual, menstrual, is referred to as the entire cycle. The process of menses is those first 3 to 5 days where we have shedding of the endometrial lining and the initiation of the process of follicular development. But that first half of the cycle can also be called the proliferative phase, and sometimes you'll see that other than the follicular phase. You know, the follicle is proliferating. So that's just a synonymous term. And then the secretory phase is also referred to the latter half of the cycle, sometimes other than the luteal phase, because hormones are being secreted. Okay, so it takes on different names, but it's the same thing. Okay. Another effect of the high concentrations of estrogen and progesterone are they have an effect on the cervical mucus. The cervix is the junction between the uterine environment and the vaginal environment. And what progesterone and estrogen do in terms of the cervical mucus is they cause, through enzymatic, not complete degradation, but they enzymatically degrade the mucus to make it very thin at this point. Okay? And then after ovulation, it causes a thickening of that cervical mucus. Because after ovulation, if you're talking about reproduction, you want, pregnancy to be occurring or to occur, and you want the uterine environment to be a sequestered environment so that the early embryo / fetus can develop, and the uterine environment being totally, well, not totally, but as sequestered as best it can from the vaginal environment. And an effect of that is the thickening, the significant thickening of that cervical mucus, okay, to cause a cervical plug. Sometimes it's referred to as a mucus plug around the cervix, okay. And again, characteristic of the human female, we have menopause, which is the inactivation of this reproductive cycle. Okay, and that can be problematic in the human, because what is one thing that's happening during this process of follicular development / ovulation? There's a lot of estrogen being produced. Okay. A significant increase in the pre-ovulatory period and a maintenance of that estrogen in the post-ovulatory period. And estrogen doesn't just play a role in reproduction. It plays other important roles, and one of them is bone development. And you've heard of osteoporosis being a not uncommon thing in postmenopausal women, because the primary source of estrogen is lost, because there is no follicular development. So there is, in effect, very low estrogen levels. And so it's not uncommon to hear about postmenopausal women undergoing estrogen replacement therapy to maintain that bone density through the maintained levels of estrogen. But then you also hear about the correlation of cervical cancer and breast cancer with estrogen replacement therapy. But there's been advances in that too. Again, the pharmacological industry is a never-ending process, and now there are a newer class of drugs referred to as selective estrogen receptor modulators. And there's one that binds to just the estrogen receptor that is specific for bone development. And so you don't get the total estrogen effect, and there could potentially be a decrease in the correlation with these cancers that we talked about, possibly. So, kind of got off track. These are two illustrations I put in. The one on the right's from your book, and the one on the left is the one I used. And I put this one in before I found this one in your book, so I just kept both of them in. But it tells you exactly what's going on in the female throughout the entire 28-day cycle, both from an endocrine standpoint, follicular development standpoint, ovulatory standpoint, post-ovulatory standpoint, even including what's happening down at the level of the endometrium. The only thing that's not shown is what starts this whole process, and that's GnRH. And the reason for that is, if you remember, I think we talked a little bit about GnRH release from the hypothalamus. It's released in a pulsatile fashion at different times of the day and at different times of the year, depending on what animal you're talking about. And so if you looked at it, you would see just, pulses. and that pulse frequency and starts to increase right around the time of ovulation, and then it decreases. So, it's just a very busy illustration. But what you'll see at the initiation of the cycle is a gradual increase in FSH, okay, with a spike right prior to ovulation. What that's causing is follicular development. Again, under the influence of FSH, a cohort of those primordial follicles will begin to develop, and they'll go through the primary, secondary. This is actually tertiary, and this is a secondary because we can see the follicular antrum. But primary to secondary to tertiary follicle, eventually to ovulation. And in the human female, we only see the LH spike. This is actually a little late. It's actually about day 13 we see this LH spike, and that causes ovulation on or about day 14. Okay, and then the actual oocyte will be eliminated, not eliminated, but extruded into the oviduct, the fallopian tube, and the remnants of the ovarian follicle become the corpus luteum. And so down here, from a endocrine standpoint, in the first half of the cycle, the pre-ovulatory period, we see the steady increase in estrogen. Why? Because we have more and more of these granulosa cells being produced. And then you can see at ovulation, we have a drop-off of that estrogen, but then it steadies out and remains high because we still have granulosa cells associated with that follicle. And now, at the post-ovulatory period, we see the presence of progesterone at high concentrations, because this is when we see the corpus luteum, the source of progesterone in that post-ovulatory period. If pregnancy doesn't occur, it degenerates into the corpus albicans. The source of progesterone is lost. The granulosa cells eventually degenerate, and those two hormones drop off. And what's not shown here is inhibin. Inhibin is also produced. Those hormones drop off, and it's a signal to start the cycle anew. Okay. If pregnancy does occur, like I said, we'll undergo a process called luteal rescue, and the source of progesterone is maintained for a period of time. And again, the effects of these two hormones are down here, in part, not just solely, but down on the endometrium, the inner lining of the uterus. If you remember, the uterus is divided into the myometrium, the majority of the mass of the uterus, the musculature, and it's actually 3 layers of smooth muscles, circular smooth muscles, longitudinal muscles, and oblique muscles. And when that muscle contracts, they contract in different directions because those muscles are oriented different to each other. But the inner lining, the endometrium, is also called the functional zone because it's the zone that changes over the course of the reproductive cycle. And under the influence of estrogen and progesterone, it's getting thicker and thicker and thicker and becoming more and more vascularized. The estrogen and the progesterone initiate a process called angiogenesis, production of new blood cells, and we have the upregulation of vascular beds in that endometrium, again making a space that's conducive for the implantation of that early embryo. Okay, why don't we take a quick break and we'll talk about fertilization and the early events in pregnancy. And we're getting actually close to the end of this chapter, which means we're getting really close to the end of this whole summer session. And again, I heard that. Somebody said, yeah, remember I've got tissues up here for if you feel emotional. I know you're excited about the end of the semester, but the end of the semester means the end of me. Not the end of me, but the end of this. And like I said, you know, sometimes students often will just feel emotional, so we got some tissue. Same for you, Professor Nold. I know you might get emotional too, so. Oh, there you go. And so do they. Me and Lucas are already kind of sort of planning a big blowout at my pond in my backyard. And be all business-like. So, we've talked about all the events leading up to and after ovulation. Now we have a oocyte in the oviduct, specifically, hopefully, if we're talking about fertilization, in the infundibulum, the upper region of the oviduct. And that's where fertilization is going to occur. If there's sperm down there, there's a likelihood that you're going to get sperm-egg interaction and binding. Now the oocyte, obviously, and this still, it's a cell, right? And it doesn't have the tail like the sperm does. It doesn't have any cilia that it can float around like some of the paramecium and cells of that nature. But it's got to get from the top of the oviduct, and if we're talking about fertilization, eventually all the way down to the uterine environment. And how does it get there? Well, there are prostaglandins that are being produced and secreted by the cells of the oviduct, which causes the muscular contraction. So kind of a wave-like muscular contraction down the length of the oviduct. And the cells of the oviduct are also ciliated. And cilia moves just like the mechanism we talked about, how the sperm tail moves through the interaction of the microtubules. It does the same thing. So the cilia just sit there on the cells and just have this wave-like action and cause a literal current in the oviductal fluid. And that, coupled with the muscular contractions of the oviduct, the egg / zygote, if we have fertilization, can move down progressively down the length of the oviduct. And this is a scanning electron micrograph of oviductal cells, and you can see that they are ciliated, and also we have mucus secreting cells as well. And I put in this cross section of a sperm tail, again, just to show you, or not show you, but remind you that the ultrastructure of these cilia are exactly the same as flagella as a sperm tail. And the muscular contractions of the oviduct, while it's helping the egg / zygote get down the oviduct into the uterus, it's also helping the sperm get up. Even though they're swimming by this point, it helps them move as well. So what we have in terms of fertilization is we need sperm all the way up here and waiting on the egg, waiting on that process of ovulation for optimal chances. It doesn't have to occur there, but for optimal fertilization, we need sperm up there in the upper regions of the oviduct waiting on that process of ovulation. And one benefit of having such a high concentration of sperm is that a lot of them are going to get stuck down here in that cervical mucus. That doesn't completely go away. It gets thinned out, like we said. But most of the sperm get stuck in that mucus plug, and then half of them that get through are going to take the wrong turn. Okay, so we need as many as we can up here. And so the sperm have a long way to go through a very harsh environment. Particularly down here in the vagina because it's acidic. So we need that buffering capacity of the accessory sex gland fluids of the male. But nonetheless, for optimal fertilization, it's got to occur up in the ampulla of the oviduct, which is part of the infundibulum, this upper region. And also remember that, you know, even though there's a physical space between the oviduct and the fallopian tube, it collectively, through these fimbria, completely envelop the ovary. So you don't actually see the ovary if you just looked into the pelvic region because it's covered up by the fimbria of the oviduct. And if we have sperm-egg interaction, again, there's not going to be just one sperm up there. In the human, we have to have, for fertilization, one sperm penetrating one oocyte, because we need 23 chromosomes from the guy uniting with 23 chromosomes from the female. If we get more than one sperm, it's not going to work. And so what we're going to have collectively is a whole bunch of sperm all working on the oocyte at the same time, and it's a race to see who gets in there first. The sperm have undergone a process called the acrosome reaction. If you remember when we saw that transmission electron micrograph of a sperm head, we saw the membrane around it. It's actually a double-lined membrane, an inner and an outer acrosomal membrane. And it's filled with acrosomal enzymes, hydrolytic enzymes that the sperm are going to use to penetrate the different layers of the egg to eventually get in. And what happens in the acrosome reaction is specific regions of those two membranes fuse and form basically pores. And so the acrosomal enzymes get out, and the sperm cell at that point is able to penetrate different layers of the egg until eventually one gets all the way in. Okay, and there's a whole host of different acrosomal enzymes in there. And then what happens when a sperm gets actually inside the egg, there's another biochemical reaction that occurs. It's called a cortical reaction. What you also have in the egg, on the periphery of the egg, are these structures called cortical granules. And what happens in the cortical reaction is these vacuoles or vesicles degranulate and cause what's referred to as zona hardening. And at that instant, once a sperm cell gets into the egg, that cortical reaction happens almost instantaneously, and the zona just turns into this hardened structure. And whatever sperm were halfway getting in, they're just stuck there. So it's a way of ensuring that one sperm gets into the inner regions of the oocyte itself and causes that cortical reaction. And once you have that, and also the head of the sperm detaches from the tail. And at that point you have unification of the maternal and fraternal chromosomes, and now you have a diploid cell, and now the structure is going to start mitotically dividing. Okay, and you can also see the emergence of these polar bodies. And in fact, once fertilization occurs, you're going to see the second polar body. Okay, and this is a really interesting scanning electron micrograph showing a sperm cell as it's penetrating different layers of the egg. And just this morning, I did y'all a favor. If you wanted to do some more reading on the acrosome reaction and the acrosomal enzymes, right down here, this is a hyperlink. And so if you click on it, it will hopefully take you somewhere. Maybe not. Well, if you do that on your computer at home, maybe it will, and it should take you to a manuscript that was authored by me on acrosomal enzymes. In the stallion, but mammalian acrosomal enzymes. So, what we have initially at fertilization is now it's no longer referred to as a egg or oocyte. It's referred to as a zygote, and it's going to undergo mitotic cell divisions as it's migrating down the oviduct towards the uterus, changing in size now. It's getting bigger and bigger and bigger because we're having more and more cell divisions, until eventually about on day 3 to 5, it's in a morphology referred to as a blastocyst. And a blastocyst is about a 5-day-old embryo. And this embryo is going to enter into the uterine environment and then just, migrate around for about 2 to 3 more days until it finds a place where it attaches to the endometrium and starts to penetrate and develop. Okay. And the blastocyst is composed, it's very easy to identify a blastocyst because it's got what's called an inner cell mass, and it's a mass of stem cells. It's those stem cells that have been so valuable in stem cell research, because a stem cell is a cell that if you give it the right environmental conditions and biomolecular cues, you can get it to develop into any tissue you want. Okay, but the blastocyst is composed of that inner cell mass, and then a periphery of cells called trophoblast cells. And the trophoblast cells will eventually become some of the tissues of the placenta. Okay. We'll talk about the preventing rejection of the embryo fetus in just a second. But again, this is the embryo, the blastocyst, one of which is Christopher. I just wanted to point out again that it's composed of an inner cell mass, and that's what you're seeing over here. Okay, these are all stem cells, and to a point right up in here and also down here. But then the cells on the periphery are trophoblast cells, and again, these are going to become part of the tissues of the fetal side of the placenta. Okay. And I just put that in this morning too because this is Christopher today. This is Christopher back then, one of them, and that's him today. Pretty amazing, huh? Going from there to there? Huh? Want to see it a lot? Okay, so this is an illustration showing the migration of that. Again, fertilization occurs way up here. And once we have fertilization, now we're going to start having multiple mitotic divisions all within the what was the zona pellucida, okay? And it just gets bigger and bigger and bigger. And by the time it gets to the entryway of the uterine environment, it's referred to as a morula, and then finally a blastocyst. And again, it's characterized by that inner cell mass, which are the stem cells, and the trophoblasts. And the rest of it's a fluid-filled vacuole, if you will. And it will just migrate around the interior of the uterus for 2 to 3 days and finally implant, okay, in a section of the endometrium. Usually in the endometrial body, you've heard of tubal pregnancies. That's when, for whatever reason, it occurs up here in the oviduct. That is a critical situation and needs to be taken care of quickly because the cell's dividing and there's not much room up here, and it can cause a rupture easily in that region. And obviously we're talking about we're having to terminate the pregnancy. You can't salvage it yet, but if all goes according to plan, implantation is going to occur somewhere up here in the main uterine body, usually on the lateral sides. But it's a process where the follicle actually starts to penetrate. Yes, ma'am?
So is it different than the ectopic pregnancy?
The ectopic just refers to out of place. And so we're having implantation out of place. And what happens is it starts to implant. It starts to do this, but up in the oviduct. And the lumen of the oviduct is very small. The width of it's very small. So when we have a dividing cell, it's getting bigger and bigger and bigger. It's going to run out of space very quickly and can cause that tissue to rupture if it's not taken care of surgically.
So tubal is like a type of ectopic.
Yeah.
Ectopic near the ovary. If a woman had like a Mirena or something put in and it goes through her uterine wall, there have been weird pregnancies, even like pseudo outside the uterus or in weird places. So ectopic just means outside where it should be, and most, like 97% of ectopics are tubules.
Yep.
They have something to implant into.
I think the word ectopic literally means out of place. And like we said, you know, the ovary, there is no physical attachment of the oviduct to the ovary. So, you know, it can get into the abdomen, and the ectopic pregnancy can occur anywhere, anywhere that's not the uterus. Usually, like we said, it's in the oviduct. Okay. But what happens once we find a knife saw place to land, the follicle, the early embryo will actually start to penetrate the uterine tissue, the endometrial tissue specifically. Okay. And again, you can see the trophoblasts, which are going to become the placenta, remain on the outside. It's actually this inner cell mass that starts to penetrate the layers of the endometrium. Okay, and it gets further and further and further in there, and then you can see how the trophoblast cells are going to become the amniotic sac. And it gets literally within that endometrial tissue. A lot of times it's thought of that it just implants on the side and just develops. It develops in the tissue of the endometrium, okay, not in the open space of the uterus itself, okay, and it will sit in here and just nest and develop for, in the female, a human female, 9 months, give or take, okay. The placenta is a very important structure that envelops the embryo and / fetus through the duration of the pregnancy. It is the structure where exchange occurs between mother and fetus. It's important to understand that the maternal and fetal blood are independent of each other, and they need to be, but there is exchange of maternal components to the fetus and from the fetus back to the mother. The mother's digestive, renal, and respiratory system are all working for the fetus because the fetal lungs haven't developed yet, the kidneys haven't quite developed yet, and also nutritional support comes from the mother. And so there is communication between the two, but fetal and maternal blood are independent of each other. Okay. The placenta also takes up or tissues of the placenta take up the role of progesterone production for the duration of the pregnancy. We talked about how the corpus luteum is a source of progesterone. It's a source of early progesterone, and then the placenta takes up for that. Also, human chorionic gonadotropin is also released early in pregnancy. And the human chorionic gonadotropin, as you would expect, is from the chorion, which is one of the inner layers of the amniotic sac. And it plays a role in maintenance of pregnancy. We have the continual secretion of estrogen and progesterone also from the placenta, because those granulosa cells from the corpus luteum, they have a finite lifespan. Even though we had luteal rescue, they're not there for the entire time. So they play a very important role in maintaining that pregnancy. Coincidentally, HCG, because it's produced in high concentrations early in pregnancy, is what's being tested for in the pregnancy test, early pregnancy test. It's basically an ELISA-like assay to measure appreciable amounts of HCG. And HCG in high concentrations has luteolytic activity too. So this is what's happening in the fetal development. Again, early on, we have a nesting, if you will, in the endometrial tissue. And then the structure stays connected to the endometrial tissue, but then the fetus itself starts to develop within the amniotic sac, again, a fluid-filled cavity, and that continues to grow through development until we get to full term, where you can see normal presentation is upside down with the head pressing against the cervical canal. We always have an association of fetal and maternal blood, but never a direct mixing of fetal and maternal blood, if you will. Because blood type is genetically determined, and the fetus oftentimes has a different blood type. And if we've got a mixing of those two bloods, we'd have a serious reaction. Okay, but there is transfer of maternal components, nutritional transfer. There is transfer of maternal antibodies. Remember, we talked about the immune system. We talked about the innate and the adaptive immune system. This is where we get the first of our the emergence of our innate immune system, because we get IgG antibodies from mom that cross the placenta. And obviously gas exchange occurs readily across these membranes, because these gases are nonpolar and they can move right across plasma membranes. So the respiratory gases are exchanged without any problem. So there is fetal communication between mother and fetus in terms of the placental structures and the circulatory systems to a point, but there is no direct interaction of maternal and fetal blood. And you can see some of the functions of HCG. Maintains that corpus luteum, although the source of progesterone, the major source of progesterone is from the tissues, some tissues of the placenta itself. You can see in the male, another important function of HCG is it stimulates that testosterone secretion from the male embryos, the ones with the Y chromosome, with that sex-determining region of Y gene on the Y chromosome. Estrogen will continue to stimulate the development of the endometrial tissue. Also, something we haven't talked about was it starts to stimulate the mammary glands in the breasts to develop, because they are going to be the source of milk. Progesterone also further develops the endometrial tissue, and at the level of the myometrium, the muscular layer of the uterus, which is the most of the mass of the uterus, it inhibits uterine contractions, because we want everything in there quiescent in terms of muscular contractions until about the 9th month. Okay, promotes formation of that cervical plug we talked about, and also stimulates development of the mammary glands. Human chorionic somatomammatropin, that's a hard word to say, but this has remember we talked about a glucose-sparing effect of some of the hormones in our body, like cortisol, things of that nature, which will direct other cells that can use something other than glucose for cell respiration to do that, so we can save glucose for some tissues. This human chorionic somatomammatropin does that. Also, collectively, is helping development of the mammary gland. So these three are synergistic hormones in terms of mammary gland development. Relaxin, this is released towards the end of gestation, and it causes a relaxation of that cervical tissue. Also starts to break down connective tissue structures, preparing for pregnancy. And then we also have placental parathyroid hormone-related peptide, which has a role in elevating calcium levels for the developing fetus, primarily for bone development. Okay. There's something else I wanted to say, but I can't remember what it was. Can't remember. I think I'll get to it. So, again, no direct interaction of maternal and fetal blood, but definitely an exchange. An important compound for our steroid hormones, remember, is, lo and behold, cholesterol. This is one thing that will be transferred to the placenta. And the cholesterol, again, is a source of these sex hormones we have in our body, and the first metabolic breakdown product in the hormone biosynthetic pathway is progesterone. So it's another source of progesterone for the female. Also dihydroepiandrosterone, the precursor to testosterone, and thus estrogen is produced, which will give us the estrogen, which also serves as an additional source of estrogen for the mother. And then in the fetus, the cholesterol being the source of those sex hormones that we talked about, beginning with after progesterone, DHEA. So there is a communication between mother and fetus, but not a direct interaction. And also, when something else that's not showing there is the maternal transfer of antibodies. Okay? And so as the process of gestation continues, the demand on the mother is quite high. From a nutritional standpoint, from a renal standpoint, because the fetal kidneys aren't really up and running at this point. And what did I leave out? Digestion as well. So it takes a big toll on the mother the further we get into gestation. Obviously, the uterus isn't really enlarging. It's stretching. So it gets a lot larger from that standpoint, but it's the fetus that's causing all this. And then as we get towards parturition, we get dilation of that cervix because that fetus is pushing down on it, starting to cause myogenic contractions. The smooth muscle of the uterus starts to contract, and you get those gentle, if you will, initial contractions until we get the initiation of that positive feedback pathway, where we start to get subtle uterine contractions, and that stimulates the hypothalamus to produce and secrete oxytocin from the posterior pituitary, and then that starts the muscular contractions that continue in this positive feedback pathway, and it just gets stronger and stronger and stronger and stronger until the stimulus it goes away, and when the stimulus goes away, what that means is we've had birth, okay, and we no longer have that uterine contraction, and the feedback pathway is relaxed. One thing I didn't point out was another hormone that we have from the hypothalamus, and thus the anterior pituitary. It starts with CRH, corticotropin-releasing hormone. And if you remember from our endocrine chapter, what this does is cause the release of ACTH from the anterior pituitary, adrenocorticotropic hormone, which has an effect on all the three layers of the adrenal cortex. And aldosterone was a big one. And it has this effect to a point, but again, the fetal kidney's not up and running as it will be in the after parturition occurs. But what CRH and ACTH do in the fetus is they collectively stimulate the development of surfactant, that inner coating, that phospholipid-rich coating inside the alveoli. And this happens right around the time of birth, so the fetal lungs haven't fully developed. And again, it's a problem in premature children because the lungs haven't fully developed in terms of surfactant. So it has an effect on gas exchange. And also there is, you would think there would be a massive antibody reaction, an immune reaction with the fetus and the maternal system. Because even though there is no mixing of maternal and fetal blood, we talked about how there is a maternal transfer of antibodies. So there is a way for the immune system to interact with the fetus. So you would think there'd be a massive immune reaction, because what you're talking about when you're having a developing fetus is a different organism, right? Genetically, completely different from the host. So you would think there'd be a massive immune reaction, but there's an actual inhibition in the inflammatory response. And it starts with the production and release of NF-kappa B, which is a inflammatory transcription factor, which initiates the activation of specific genes. One for IL-8, it's one of the interleukins, an inflammatory mediator, as well as prostaglandins, which are have multiple roles, but one big thing is they can cause smooth muscle contraction. But what this specific type of inflammatory mediators, they're actual ones that inhibit the immune response to a point. One of the inflammatory mediators is a special type of, I'm drawing a blank, regulatory T cells that actually, if you remember, regulatory T cells do that. They regulate the immune response, and one of the partial regulatory roles is they dampen the immune response. It causes the production of these types of regulatory T cells, which keep the immune response in check, so there is no identification of this fetus as foreign tissue. And this busy slide is just the whole sequence of events that initiates birth, okay? And it starts with those subtle contractions of the placenta and the initiation of the release of CRH. This causes the release of ACTH, which are going to have an effect on the fetal lungs, the beginning of the fetal lung development in terms of surfactant release. It's going to also be converted to the DHEA, the dihydroepiandrosterone, which is going to give us our source of estrogens and progesterones, which is going to increase the estrogen concentration. And over on the fetal side, again, we're going to have development of the fetal respiratory tissues with the production of surfactant. The initiation of macrophages, or activation, I should say, again, and that's through the activation of that NF-kappa B, which is an inflammatory transcription factor. So we'll get the increase in IL-8, IL-8 beta specifically, which we get a further activation of NF-kappa B in the uterus, and release of progesterone from the corpus luteum. Eventual beginning of the release of oxytocin and prostaglandins. Both of these are going to cause a muscle contraction, so now we're starting to get contraction of the uterus. This oxytocin is in a positive feedback pathway with the maternal side, and we're going to begin to get those uterine contractions again and again and again, stronger and stronger and stronger, because we're getting the release of oxytocin from the posterior pituitary, and it's in a very tight positive feedback pathway, whereas we continually have contractions of the uterus, we have continual signaling for more oxytocin to further enhance that process of smooth muscle contraction, all coupled in to the release of prostaglandins as well, and that will continue until we have expulsion of the fetus, okay? And an elimination of that cue for the feedback pathway of oxytocin. Okay. We also have, at the level of the mammary glands, the process of lactation. And initially mediated, and specifically referring to the development of the mammary glands in the breast. Progesterone and estrogen are the two major hormones in that regulatory pathway. There are others, but those are the two big ones that cause the development of those mammary tissues. And that, along with prolactin, also from the anterior pituitary, stimulates the production of milk in those mammary glands. And that continues throughout the gestation, most of it occurring towards the end of that parturition period. But the milk is maintained in the mammary glands themselves. And it's not until we get the release of oxytocin. Oxytocin, another secondary effect of oxytocin, is called milk letdown. And it causes the release of that milk from the mammary glands. And that's why sometimes late in pregnancy, the mother will have milk ejection from the breasts before she's given birth, but that's because she's starting to get the emergence of that release of oxytocin, which is causing milk letdown from the mammary tissue. Okay, and so again, normal presentation is head first in the human and the foal. Development of the breast occurs specifically at the level of the mammary glands. So the breasts themselves don't get bigger, but the mammary glands do. Okay, and it causes an enlargement of the breast because they become engorged with milk, and it's not until the release of oxytocin that the milk is actually ejected from the mammary glands. And that's also an example of a positive feedback pathway. As long as the female is breastfeeding, allowing the baby to breastfeed, you will have continual stimulation of the release of oxytocin that causes the continual release of milk from the breast. So as long as you get that stimulus, lactation will continue. And you've probably heard or know of some people that are big breastfeeding fans, and I am too, but they'll allow their children to breastfeed until it just gets kind of creepy. I remember my ex-cousin, she just thought breastfeeding was great, and it is. It's very important. But she would let her children breastfeed way beyond. I remember one time I went to dinner at my in-laws' house, and Alexander wanted to breastfeed, and I don't know how old he was, but he just started reaching up and unbuttoning her shirt. Huh? I've seen that, but I can't remember. That was like the second one, Meet the Fokker, yeah. Oh, yeah. Yeah, so yet another example of positive feedback pathways, which can be negative in nature, depending on how you look at it. And that's the end of the chapter. Points to ponder. Why do you think most oral contraceptives are for women? Why don't they make them for men? Make the men take care of the problem. There's definite, it's a big area of research. The female, the pill is just a dose of estrogen and progesterone. So it utilizes that negative feedback pathway. And so if you maintain high levels of estrogen and progesterone, you're going to inhibit FSH and LH. You don't have follicular development. Problem with the male is there's no cyclical changes in our hormonal system. We produce testosterone, and that's what drives the whole process. And you could effectively inhibit that, but testosterone has a lot of other important beneficial functions. Protein synthesis is a big one. So it's a little bit more difficult, but they are making, that's a big area of research for male oral contraceptives. And you know, that video I showed you, you know, the Africa thing, that was a contraceptive we were using in animals, and it was utilizing the animal's immune system to produce antibodies against the zona pellucida to block. Remember, the zona pellucida is made up of 3 glycoproteins, ZP1, 2, and 3, and ZP3 is the sperm binding protein, and if you can block that, you can block effectively sperm binding. So that's another form of contraception that's a big area of research. We talked about the vascular mechanism of erection and how does Viagra help. So that's it for the whole summer session. What I thought I would do on our very last day, Friday, is you know, review for the exam between then and now.